the Nature Conservancy is a nonprofit organization that works around the world on habitats. And we have been using drones for probably 6 years now, 6 or 7 years. Actually, since May 2014, so I guess almost 10 years now. And we're really interested in leveraging this technology and being able to build capacity with the partners that we work with. Our goal is to help countries protect their habitats and preserve life preserve these habitats upon which all life depends. And we've been able to leverage this technology specifically in our coral monitoring work. We spend a lot of time growing corals in nurseries and outplanting those coral. And we're interested in identifying how that restoration work is succeeding. And so I'm going to walk you through kind of the history of how we've been using drones over the years, and specifically show you some of the results from our deep learning algorithms, where we feed this imagery into a deep learning environment and try to detect the occurrence of these coral colonies. So, back in May 2014, we started with this drone, which we built. It was built on a budget of $2,000, and we used a GoPro and had a custom 3D model frame with all those electronics inside of a Tupperware that you see there. And our goal was to, as we survey, we wanted to get into areas where we couldn't access with the boat, that were shallow. And we were creating a lot of benthic habitat maps from both satellite imagery and drone imagery. And this was kind of our first prototype. And then we moved over the next year into a 3DR Solo platform, where we used modified GoPros to be able to capture both RGB and infrared. So we were trying to find the latest and greatest platforms to do this work. We did move over to a Sony QX1 camera, which provided us better imagery, acquiring 20 megapixel stills. And then we moved over to DJI products, and we're currently using a Mavic 3 multispectral, and we also use a Wingtra Gen 2 drone, which has a Sony RX1 R2 camera, which really has been incredibly helpful. I know David, he talked a little bit about fixed wing drones in the beginning, and we found that these are really helping us cover larger areas. We can fly higher, and this drone is able to map up to 50 to 60 minutes, depending on weather conditions. And with that 42 megapixel camera, we can fly higher and acquire better resolution imagery. For the coral reef monitoring work that we do, we fly at about 65 meters, and that gives us about a 1 centimeter pixel. And we've done this throughout many different areas of the world. Here's the team that I worked with in April, collecting imagery in Moorea, an island in Tahiti. We had 2 drones mapping a third of that island, and I'll show you some of the results. We originally looked at Worldview 2 imagery to be able to map these coral colonies. The reason that we were mapping this area is because there's a group called Coral Gardeners that works to restore some of the coral that's been lost in Tahiti and around the world. And they were really interested in the question of, can we map live coral? And that's a very difficult thing to do. Especially with satellites, it requires hyperspectral imagery and a lot of field data. And we wanted to ask the question, well, if we get the spatial resolution fine enough, can we identify those individual live coral colonies? And so here's an example of what the imagery from the WorldView 2 satellite looks like. And we tried to classify that, but in our initial attempts, we realized that it wasn't possible with the limited bands. And so I want to show you some of this drone imagery that was acquired with a Wingtra, flying at 65 meters with a 1 centimeter spatial resolution. You can see here the coral nurseries that are where they have these tables laid out where they're growing coral, and then they take that coral and outplant it onto the reef. And you can see as we zoom in the spatial resolution where the color indeed represents live coral. And so we wanted to take advantage of this high spatial resolution, high contextual data set to see if we could identify individual coral species. And so we worked with the team there, the experts in identifying the different species, and these are the ones that we could identify from the imagery. And so we built a machine learning model around this, trying to identify if these individual coral colonies. And so this is the imagery. We segmented the imagery, which groups similar pixel reflectance values. And then we would classify those. And we're in the process of finishing this work up and trying to assign these individual classes to coral, but the color really drives the identification of these corals, and also the texture that's found within each of the images. I want to show an example also in the U.S. Virgin Islands. This is in St. Croix, an area around Buck Island, and also a linear reef called Lew's Reef that we worked on. We collected a lot of field data. We used an underwater camera that was attached to an RTK GNSS, and then we PPK corrected that and got our accuracy down to about 2 to 3 centimeters at every photo. And we needed that accuracy to be able to really identify. You can see some of the underwater photos that were taken. So when we flew the drone imagery, we collected the RGB data, very similar to what David was talking about. We ran a Python script to extract bathymetry, and we used both of those, the RGB data sets and the bathymetry, to feed that into the machine learning. And I want to show you some examples here. So here's a coral colony. It's a Cropra palmata, or the common name is elkhorn coral. And this is a very important shallow reef building coral that experienced a lot of die-off, and it's actually an endangered species. And so we're trying to regrow colonies of these and establish the population and look at genetics. We hear a lot about bleaching and disease, and so we're really interested in growing genotypes that seem to be more resilient to some of the threats that corals are facing. And so we want to monitor their growth and collect fragments from this species, and we microfrag those. We cut them with a bandsaw, which stimulates the growth. We put those in nurseries, and then those fragments grow at an accelerated speed. And so here we can see what that colony, that exact same colony, looks like from above. And if we take different data sets, this is looking at it from using a Phantom 4 at a low altitude. This is from a Mavic 2 at a higher resolution. This is the data set from the Wingtra flying at 75 meters. We want to understand different environmental conditions. This was partly cloudy in the afternoon. This is clear in the morning. So we were collecting a lot of different data sets at different times of day and different conditions to see how that colony changed over time. And you can see if we raise that flying height up to 90 meters, what it looks like, and then going to 120 meters. during cloudy conditions. So we evaluated what these corals were what they appeared like at different conditions, at different times of day, at different altitudes, to collect a library that we could train the machine learning network on. And so here you can see some training data that was fed into the deep learning algorithm. We looked at different algorithms and tried to assess their accuracy. And we're working on a paper right now to report out the accuracy. I'm working with Dr. George Raber at the University of Southern Mississippi, and he's leading the deep learning work. But we're finding very promising results in being able to identify these individual coral colonies and be able to quantify live coral. And this is really exciting for us because we're doing a lot of outplanting of coral, and we want to measure our success. And some of the initial findings show the density of this Acropora palmata species across that area in Lewes Reef in St. Croix. We have some preliminary models. We flew it just 3 months ago, and we're comparing that data from the data set we collected 2 years ago to look at growth. And we know that coral grows very slowly, but Acropora palmata, this particular species, is one of the faster growing species. And so we're having success identifying those areas that have increased in size. We're looking at different things like, is there a difference between using a JPEG compression versus a RAW? And you can kind of see the differences going from JPEG to RAW. JPEG is a much smaller file size, but the RAW gives us a little more detail. We're looking at different post-processing algorithms that we use, for example, drone deploy or metashape, and how those orthomosaics appear and how they function in these deep learning algorithms. We're doing a lot of work mapping areas where these governments are experiencing beach erosion. And a lot of that beach erosion is due to an erosion of the coral. The coral is important because it's protecting that shoreline. It's attenuating the wave energy that comes in. And so many governments are very interested in restoring their coral. And this involves coral nurseries as well as hybrid solutions, where we work with shoreline engineers to develop artificial reefs. And then there's also hybrid solutions. And this information is really important. We can take drone imagery and we can classify where the live coral is, where the rubble and hard bottom, the sand, the seagrass, and the turf algal substrates. And so we see this as a promising area, in particular for local scale studies. Here's some work that was published that I was involved with in Belize, where we were looking at success of outplanting efforts in an area called Laughing Bird Key National Park. And we demonstrated that we could identify a 17% increase in Acropora cervicornis over time, which is the species that, and for this particular project, they're really interested in. So real quick, some of the lessons learned: we experimented with different flying heights, and we identified that flying around 65 meters was sufficient at a 1 centimeter pixel to map the individual coral colonies. Now, with that flying height, when your coral colonies are less than 50 centimeters on a side, half a meter, our accuracy goes down. So once it gets above that threshold, our accuracy exceeds 80%. These smaller colonies are a lot more difficult to detect because they get confused with other features, the sea fans and other species of coral that are similar, that have a similar color. And we also determined that we can detect about a 5% change in the coral once it reaches that 50 centimeter threshold. And it's very useful to acquire higher resolution mosaics for monitoring these small areas because it really helps with the selection of the training data. And also, the field data is very, very important. So some of our research areas that we are exploring is what are the ideal times? And these are some of the things we're addressing in the paper that we're working on, looking at cloud cover, sun angle, water motion. These data sets have to be acquired at low sun angles, typically early morning or late afternoon, to avoid things like sun glint. We're looking at different camera parameters under different sky conditions, looking at shutter speeds. We want to avoid image blur, and we want to also capture that information at its full content and contextual information. And so that means experimenting with different ISO levels. We want to look at different post-processing softwares. What are the pros and cons of each, and what produces the best output for deep learning? How does depth or distance from shore relate to coral fragment growth over time is another question we're trying to answer. And the flying lap and mission parameters that we set, how does that affect the ability to stitch these orthomosaics, and how can we use color correction to improve detection performance? So those are some of the questions and ideas that we're researching. We're talking about drones, but I want to take you underwater and show you some of the work that we're doing with close proximity photogrammetry. So here's an example of a camera system that's set up that's on a boogie board where we're collecting stereo photo. We have a snorkeler that's pushing this along in a lawnmower pattern, and we're collecting stereo data just like a drone. And we can also do this via scuba, and we can get 2 meters from the reef, and we can collect stereo images, very, very detailed stereo images of the reef. And this captures information that we can't answer from the drone imagery, because we know from drone imagery our ability to detect features that exceed about 2 to 3 meters is difficult because of the refraction of the water and sometimes the turbidity or the visibility of the water column. So we collect these data sets much like we do with a drone. We're doing this in typically 10 by 10 meter monitoring plots. We put down scale bars, and we put in markers, permanent markers, into the coral that allow us to be able to line these data sets over time and do very detailed change detection. We also do a lot of pre-processing color correction because we know that the different colors of wavelengths are absorbed the deeper you go. And so we try to introduce, there's methods to introduce that those reds and those greens back into the image data sets, which helps with the identification of these species. Here's an example of some outplants on a coral. This is in Bahamas, where we have cervicornis, a proper cervicornis growing, or this staghorn coral you can see. And this information really helps us identify a lot of indicators that traditionally scuba divers have collected, but we can do this with much detail at a finer resolution. We can do it for these monitoring plots and calculate things like spatial ecology, the structural complexity, the demographics of coral colonies over time. Very, very, very powerful way to monitor these coral reefs. We can bring it into software such as TagLab, which is an open source software to be able to label and identify live coral, the presence of disease, presence of predation, and track these over time and see how these reefs are changing. So this is something that we're doing with the Perry Marine Institute in areas across the Bahamas. And some of the challenge are getting accurate enough georeferencing information to be able to align those pixels over time, dealing with data gaps and having sufficient overlap. analyzing 2D versus 3D models and trying to get that side information of the coral when you've got high rugosity, looking at water quality issues, backscatter, sunlight refraction is all issues that we have to deal with in the water column, the sheer data volume, processing power, and then the capacity of being able to extract the information that we need and build that skill set with the partners that we're working with. That's another challenge that we have. So with that, I'm going to go ahead and end.